These are the summaries of the all of the antibiotic drugs, but now I will focus on one class and I will tell you that in short, right? It's a 3 minutes video, so focus here for the GPAT. So anti-metabolite, like we use in cancer, anti-metabolite drugs, right? Anti-folate, purine, anti-pyrimidine, but those drugs in that case which are anti-cancer are not used as antibiotics because they are not specific to the bacteria. They can bind to the human, right? But here these drugs which are used as anti-folate, as antibacterial drugs, are the sulfonamides and these trimethoprim, which are anti-folate, right? Anti-folate drugs. So they help in the inhibition in the formation of the folic acid tetrahydrofolate with because this folic acid will help in the formation of the purine and pyrimidine, and more specifically if talk about the purine synthesis of guanine, right? Hope you got this one. Now talking about these anti-metabolite drugs, hope you know that what is the difference between. Now maybe you have some questions like methotrexate is a anti-folate drug, can't we use it here? No, we can't use because it binds you generally with the human DHFR, right? Human, right? But this only inhibit the tetrafolate or folic acid only and only in bacteria. Hope you got it. That's why it is as an important antibacterial drug, right? And it is a bacteriostatic, right? Both. So here is a process of this is a PABA, para-amino benzoic acid, is then converted into the dehydropatroate with the help of the enzyme dehydropatroate synthase enzyme. Then we have DHFS, dehydrofolate dehydrofolate synthesis. So which is this dehydrofolate, इधर हमारा क्या बन जाता है, dehydrofolate बन जाता है. Then it is converted into the dihydrofolate से convert हो जाता है tetrahydrofolate में with the help of the enzyme dihydrofolate reductase enzyme. This is the main product tetrahydrofolate which is used in the nucleotide, right, amino acid synthesis and all DNA synthesis, because the basic is the DNA synthesis. If DNA is not synthesized, then it cannot be converted into the RNA, and then it can't be decoded into the amino acids and can't make any protein, right? Hope you got this process. So the two drugs which are used here is the DHPS, dehydropatroate synthesis. जो पहला स्टेप है ना, पाबा से लेके ये डिहाइड्रोपेट्रोएट बन रहा है, इसी में ब्लॉक करने के लिए हम यूज़ कर रहे हैं क्या, DHPS एंजाइम डिहाइड्रोपेट्रोएट सिंथेज. सो हियर वी आर यूजिंग sulfonamide drugs and in the last step of the enzyme. So remember we are using here in the first step and very first enzyme and last step. बीच में हमारा कुछ भी यूज़ नहीं हो रहा है, ठीक है ना. डिहाइड्रोफोलेट के बाद कुछ नहीं हो जा, यहाँ पे हमारा हो रहा है, मतलब यहाँ पे यार जो ये है ना इधर, बट हमारा जो लास्ट स्टेप है ना, जो एंजाइम हमारा टेट्राहाइड्रोफोलेट बना रहा है, उस एंजाइम को हम क्या करेंगे, इनहिबिट कर देंगे. सो बाय इनहिबिटिंग द DHFR डिहाइड्रोफोलेट रिडक्टेज एंजाइम वी विल प्रिवेंट द सिंथेसिस ऑफ टेट्राहाइड्रोफोलेट. एंड विच ड्रग इट इज ट्राइमेथोप्राइम, विच इज ट्राइमेथोप्राइम DHFR एंटागोनिस्ट. होप यू गॉट दिस वन. And there is a combination given very potently which is cotrimoxazole, which is a potent and very useful question in an exam. So what are the drugs in the combination are used? It is not a drug, it is a combination of drugs, a combination known with this name. So it contain sulfamethoxazole and trimethoprim, the two drugs which are used simultaneously in a combination and to prevent the folate synthesis complete in a potent manner, right, in a potent manner. So एक ही साथ हमारा ये भी ब्लॉक होगा और ये भी ब्लॉक होगा, ये तो कैसे ही बन जाएगा, इन बैक्टीरिया ओनली. थैंक यू गाइज, थैंक्स फॉर वॉचिंग.